So there's a good bit of confusion on how to draw the complementary strand of a base pair of DNA. So right now I'm going to show you how we would draw in those backbones. So on the left we have the parent strand, which is 5' to 3', because we start with the 5' phosphate up here, although we can only see the oxygen for the 5' phosphate, and we end with the 3' hydroxyl. And you'll see on the opposite strand, I've drawn in the bases and I've drawn them in the opposite conformation. And so what you can do when you're drawing in the complementary strand of DNA is you can draw in the bases and then work in the backbone. It's going to look a little awkward, that's true, but I'm not so much worried about aesthetics as that you understand what's going on. So one of the things we can do is we can draw this bond a little bit at an angle. So we'll go ahead and we'll erase the straight up and down bond, the glycosidic bond from the base to then that nitrogen. And we remember that that's where the base is attached to the ribose. So here we have the ribose ring in the same direction, or the oxygen at the top of the ribose ring is pointing up. So what we can do is we can just point the ribose in the opposite direction. So there we go. Look to the other side. And again, it's not the cleanest drawing, but it's something. If I were in ChemDraw would be yelling at me, but that's one way. So we've got the ribose now. We got the riboses drawn in. And again, they're flipped 180 degrees for how we would draw them on the parent strand. So we've got these, so we know that if we're going to 5' to 3' in this direction, we have to go, 5' to 3' in the opposite direction. And that's when we flip the riboses up. So we have to start with the 5' phosphate up here. So we'll go up and over, draw the oxygen, and we'll draw a little squiggle there to signal that there's another strand. But if we were the rest of the strand, but if we were to draw it in, we would draw it in like so, and we could draw in a squiggle over there. Okay, so that's the 5' phosphate. Now we have to draw in the 3' hydroxyl. This is DNA, so we don't have a 2' hydroxyl. So we've got the oxygen, but we've got another nucleic acid, another DNA base coming after it. So we actually have to draw in the phosphodiester bond. Suppose in this case we would draw the P upside down like this, but it really doesn't matter as long as you have the P in there. We have that oxygen, and then there's another carbon, and then it links to the next ribose, deoxyribose, or you could draw it like that. Either way, just to indicate that there is a CH2 between the oxygen and the ribose ring. Okay, and then we would draw the 3' hydroxyl over here, but so in this case, actually, if we go back here, we would end at the phosphate because this is the end of our parent strand, so we would end with a phosphate on the opposite strand, whereas on this strand, that would go and form a phosphate to the next base pair that would be down the sequence on the other side. So you can see here to draw in the complementary strand, where we end at where we have the hydroxyl on one side, the end hydroxyl, we have the end 5' phosphate on the opposite strand, and then on the parent strand, where we have the connected 5' phosphate, we would have the connected 3' hydroxyl on the opposite strand. And then to make sure this works properly, you can draw in the bases the same, because this is a flexible glycosidic bond. It's flexible, all right? And the bases can rotate around. But in order for the structure to work out and to have things going in the proper orientation, we would draw the ribose rings as flipped 180 degrees as to how they were on the parental 5' to 3' strand.